Myself, Dr. Roma Ghai, Associate Professor from Kite School of Pharmacy, Kite Group of Institutions, Ghaziabad, would like to discuss the topic Vaccine Pharmacovigilance and Vaccination Failure. Now, this particular topic belongs to the branch Pharmacy and the subject Pharmacovigilance PP 805ET. So, let us start the introduction. Now, as we all know that vaccines play a very pivotal role in modern healthcare delivery. Pivotal means very important role. Now, vaccine as per WHO definition is a complex biological preparation which functions by inducing the immune system of the body to elicit an immune response to a particular pathogen. So, vaccine is simply a complex biological preparation and simply it is producing an immune response to the body, and that immune response specific to a particular pathogen. Now, vaccine contains an agent and that agent resembles a disease causing microorganism, and it is made from either weakened form or the killed form of the microbe or its toxins or its surface proteins. Now, vaccination is the process of administering the vaccines is called, the vaccination is the process of administration of antigenic material, that is we are administering an antigenic material to stimulate an individual immune system to develop an adaptive immunity response to a pathogen. It is one of the very most effective ways to prevent diseases, and over the years vaccination has prevented many serious illness, many serious disabilities, and has saved millions of lives across the world. Now, there are different types of vaccines. I would like to tell you about the different types. Number one is live attenuated vaccine, second one inactivated vaccine, third is subunit vaccine, then we have toxoid vaccine, and then we have the viral vector vaccine, just like Covishield. Now, I will be coming one by one in all these vaccines. Now, different types or different formulations will affect how these vaccines should be used, how they should be stored, how they should be administered. If we want safe and efficacy from the vaccine, it is vital to be familiar with the different types of vaccines and how to handle them. Different vaccines can cause different areas, and it is very important to recognize what these may be. Let us see one by one. First, we have live attenuated vaccine. The vaccines examples are LAV vaccines, examples are TB, that is BCG, which we give for prevention of TB, oral polio vaccine for prevention of polio, then there is measles vaccine for prevention of measles, rotavirus vaccines, yellow fever vaccines, varicella zoster vaccines, and seasonal influenza, which can come as nasal spray. So, all this category comes under live attenuated vaccine. The next one is inactivated, that is containing killed antigen. Examples are whole cell pertussis vaccine, inactivated poliovirus vaccine, hepatitis A vaccine, seasonal influenza injectable vaccine. Next, we have subunit purified antigens vaccines. Examples are acellular. When we say acellular means there is absence of cell. Acellular pertussis vaccine, Haemophilus influenza type B vaccine, pneumococcal vaccine. This is PCV 7, PCV 10, PCV 13. PCV means it is basically conjugated, pneumococcal conjugated vaccine. That means this is effective against 7 serotypes, this is effective against 10 serotypes, against 13 serotypes. So, these are the subunit vaccines, 13, 10, 7. Okay. Then we have hepatitis B vaccine, right? Then human papillomavirus vaccine, that is HPV vaccine. Then we have toxoid vaccines where there is inactivated toxins. Example, tetanus toxoid, diphtheria toxoid. Then we have viral vector vaccine like Zaira Ebola virus vaccine. So, let us start the first category, LAV vaccine. Now, it is available since 1950. It is derived from disease causing pathogens. That means either we are using live virus or we are using live bacteria. Now, this live virus or live bacteria has been weakened. That is why it is called attenuated. That is why the word is come attenuated. So, weakened under lab conditions, and this produces excellent response, right. Now, live microorganisms provide continuous antigenic stimulation and it gives time for memory cell development. Attenuated pathogens can revert to original form and cause disease. So, this is a drawback of these vaccines. Sometimes the weakened form can convert into a original form. They can harm individuals with compromised immune system. So, we should not give them to the compromised immune system. We should not give in AIDS. We should not give them to pregnant women. They are less safer as compared to inactivated vaccine. If we see the next category of vaccine, that is inactivated vaccine. Now, they are made from microorganisms, viruses, bacteria, just like the LAV vaccine. However, in LAV, the live virus or bacteria is attenuated, that is weakened form. Here it is killed, that is the virus bacteria have been killed through physical or chemical processes, and these killed organisms cannot cause disease. So, they cannot revert to active forms. Then type of immune response they produce, they may not always induce an immune response, and response may not be long lived, So, we have to give some more doses. Several doses of these inactivated vaccines may be required to evoke a sufficient immune response. Inactivated whole cell vaccines have no risk of inducing the disease because they are killed forms, right. So, I am sorry for the spelling here, it is inactivated, it is killed antigen, right. So, inactivated whole cell vaccines have no risk of inducing disease as they do not contain any of the life components. So, they are more stable as compared to LAV vaccines. Let us go to the next type of vaccine, that is subunit vaccine, as I have already told you the examples. Now, these do not contain the life components just like inactivated, but here it is subunit, that means that it contains only the antigenic part of the pathogen, it only contains the antigenic part, and these parts are important to elicit means produce a protective immune response. Now, these subunits could be protein based, polysaccharide based, conjugates. So, we may not go in detail of the vaccine because our focus should be on vaccine pharmacovigilance, not in development of vaccine. So, subunit vaccines do not contain the life components. So, they are very, very safe, no risk of inducing the disease, safer, more stable than LAV. Then we have toxoids. Now, toxoids is nothing called, it is called inactivated toxins. That means the toxin has been inactivated through some mechanism. So, that is the protein based toxin is rendered harmless and it uses as the antigen in the vaccine to elicit immunity. So, that means the protein based toxin is used as antigen. To increase the immune response, this toxoid is adsorbed to aluminum or calcium salt which serve as adjuvants for the vaccine. Now, they are very, very safe because they cannot cause a disease, they cannot cause a disease which they are preventing, and there is no possibility of reversion to virulence also. Again, they are stable, less susceptible to changes in temperature, humidity and light, so you can store them easily. Examples are tetanus toxoid and diphtheria toxoid. Next one is viral vector vaccine. So, we have two examples, that is a recombinant, this particular is vesicular stomatitis virus, and this is Zaira Ebola vaccine against Ebola. And second example is COVID vaccine. This is a well established technology, it produces strong immune response involves B cells as well as T cells. Previous exposure to vector can reduce effectiveness. If we are exposed to the vector, it is relatively complex to manufacture. They are relatively complex to manufacture. So, what is the difference? They do not actually contain antigens, but rather they use the body's own cells to produce them. They use the body's own cells to produce those proteins. How do they do? They use a modified virus to deliver the genetic code for antigen. Here, for example, modified virus, like in COVID, there is adenovirus, COVID vaccine, adenovirus was used, right. So, you use the modified version to deliver the genetic code for the antigen. So, by infecting the cells, instructing them to make these antigens, so antigens are made inside the body and these antigens will trigger the immune response. So, the vaccine mimics what happens during natural infection. जो natural infection के time पे होता है वैसे ही actions होते हैं by antigen. So, this has the advantage of triggering a strong cellular immune response by T cells as well as production of antibodies by B cells. So, we have seen most of the type of vaccines. Now, coming to our topic, vaccine pharmacovigilance, we all know that vaccines are the best defense as we have against infectious disease, but we cannot say that no vaccine is 100% safe for everyone. No vaccine is 100% safe effective because every person responds to vaccine its own way. Vaccines, however, not risk free, adverse events can happen occasionally, and to ensure that to ensure continued success of vaccines, we need to watch the adverse events for vaccines. It is crucial to make sure that vaccine should be safe. So, according to CTOMS WHO working group, we call that this vaccine pharmacovigilance is defined as science and activities relating to the detection, assessment, understanding and communication of adverse events following immunization. So, vaccine pharmacovigilance deal with all the signs and activities related to the detection, assessment, understanding, communication of all adverse events following immunization and other immunization related issues, as well as prevention of the untoward effects of the vaccine. So, what is the need? Need is very important. We have to promote the safe use of vaccines among the recipients who are receiving it. Early detection of adverse events is very important. Vaccines are given to healthy people. See, the person is healthy and we are giving vaccine. So, healthy persons are they are already healthy. They will feel that if vaccine is wrongly given to them, they can produce harm. So, we have to ensure that vaccines are safer. Most of the vaccines are administered to infants and children. We have seen that most of the vaccines are given to infants, like BCG is given to infants, DPT is given to children. Vaccines are given globally to large populations. Vaccines are administered concomitantly with other vaccines. That is, not only one vaccine is given, many other vaccines are given. Vaccines are complex biological products. So, our goal of vaccine pharmacovigilance is to detect untoward effects of vaccine and appropriate timely response to any adverse event. If some adverse event happens, then we have to respond in timely. So, we want to actually minimize the negative effects to the health of individuals. So, continuous risk, benefit assessment and risk management are integral part of vaccine pharmacovigilance process. So, what are the objectives of immunization safety? First, very important objective is to detect and timely identify the problems. We are identifying the problems with vaccines, which could be due to their inherent properties, quality defects. So, we can identify the problems with vaccine lots or brands which have led to the vaccine reactions. Then next objective is to detect, correct and prevent immunization errors. Sometimes errors occur due to errors in vaccine preparation, handling, storage or administration. To ensure that coincidental events are not mistaken for vaccine reaction. Then we have to identify signals of unknown vaccine reactions to generate new hypotheses about vaccine reactions, which may be specific to a particular population. To determine observed vaccine reaction rate and then related with other country vaccine reaction rate. To reduce the incidence of injection reaction that is caused by anxiety or pain. So, in that case we have to reassure the public that the vaccine is safe. Other objectives: to collaborate and then share information between the National Regulatory Authority and immunization program and other stakeholders to ensure vaccine safety. So, vaccine pharmacovigilance is nothing but ensuring vaccine safety. To collaborate and share information with the WHO regional office and Western Pacific and globally to generate new and additional information on vaccine safety. Now, there is high level of safety required for vaccines. What are the elements to be considered when we are conducting vaccine pharmacovigilance? We should remember that vaccines are given to healthy people. Vaccines are given to infants. Vaccine may be administered to the vast majority of population or a birth cohort or maybe groups at high risk. Sometimes we give, suppose a person is having some elderly persons, they are recommended pneumococcal vaccines. Why? Because they are at high risk for pneumonia, right. Subpopulations may be more susceptible to experience certain adverse events following immunization. The age at the time of immunization may coincide with the emergence of certain age-related diseases like neurodevelopmental disorders. Immunization for certain vaccines is mandated in some countries. The benefits may not be immediately visible. The benefits may not be this, particularly if the target disease incidence is slow. Now, vaccine fails. Why? When it fails? The vaccine failure occurs when disease occurs in a person despite he is being vaccinated. Now, we have number one is primary vaccine failure, the other one is secondary vaccine failure. So, primary vaccine failure occurs when when a person fails to produce antibodies at a detectable level, एक level होना चाहिए antibodies का, तभी immune response produce होगा. अगर वो fail हो गया है, the person is not able to produce enough antibodies, he is not able to respond to the vaccine, then we call it primary vaccine failure, because the protection from the disease is does not come inside the body. The second one is secondary vaccine failure. This is when the person does produce antibodies. Antibodies are produced in response to vaccination. However, the levels wane. Wane means they go down. They decline at a faster rate than normally expected. However, antibodies to almost all vaccines decline over time. We should know that is why the boosters are given after some months, because the antibodies decline over time even after booster shots, and especially secondary vaccine failure and outbreaks of disease is frequent amongst the vaccinated, is frequent. हमने जब आपका कोविशील्ड और कोवैक्सीन आया तो हमने देखा बहुत लोगों को कोविड हो रहा था. क्या प्रॉब्लम आ रही थी? सेकेंडरी वैक्सीन फेलियर. उनके जो कोविड जिनको हुआ था उनकी एंटीबॉडीज बिल्कुल डाउन हो रही थी, राइट? तो सेकेंडरी वैक्सीन फेलियर, so maybe this could be the reason, right? So one is primary and the one is secondary. Let's see next one, causes of vaccine failure. What are the causes of vaccine failure? Why was this vaccine failure? Maybe some vaccines, they are maintained only in chilled condition, cold condition. Maybe the cold chain maintenance was not there. Maybe poor vaccine storage. Maybe inappropriate vaccine transportation. Maybe poor water quality for reconstitution of vaccine. Maybe exposure to UV light. Maybe poor vaccine condition. Wrong timing of the vaccination. Incorrect vaccination technique. Improper duration of vaccination. Health status. Presence of respiratory disease. Immunosuppression, inappropriate choice of vaccine, poor responsiveness in allergic and obese persons, as well as aging. These are some of the causes of vaccination failure. Now, vaccine pharmacovigilance, vaccine safety is critical to the success of immunization program. If we want our program to be immunization program to be successful, so vaccine safety is very, very critical, right? Now, if you see this WHO vaccine safety basics, most of the material has been taken from that. So, our general public has low tolerance to adverse events. Obviously, the public is healthy. Why the person should take vaccine if he is not sure about vaccine, that vaccine can produce any type of harmful effect also. So, general public has low tolerance to adverse events as vaccines are given to healthy people. So, there is expectation to safety standards is also higher as compared to medicines. So, vaccines have the people expect कि उसकी safety ज़्यादा होनी चाहिए. So, national regulatory authorities have ensured quality, safety, effectiveness of vaccines. So, before being introduced, they are assessed in clinical trials. Once they are introduced in the market, they are continuously reviewed, and the national regulatory authorities are then monitoring as well as investigating any type of adverse event following immunization to ensure safety for the population. So, just same thing, higher standard of safety is expected, and since that's administered to a large number of healthy people, goal is prevention of disease, not treatment. So, we have the vaccine pharmacovigilance focuses on that. Then there is low risk tolerance. Now, vaccine in 2011, WHO had developed a strategic document on vaccine safety called Global Vaccine Safety Blueprint. This document sets out indicators that ensure that all countries have at least a minimal capacity to ensure vaccine safety. So, this blueprint proposes a strategic plan for strengthening vaccine safety activities globally, right. So, this is the source of this particular picture. So, some elements are directly related to vaccine system, like we have to strengthen the vaccine safety monitoring. The objective is strengthening the ability to evaluate vaccine safety signals, develop vaccine safety communication plans, understand the perception of risk, prepare for managing any AEFI and crisis promptly. Develop internationally harmonized tools and methods for vaccine pharmacovigilance. So, this is directly relating to vaccine system. And this initiative supporting elements are establish a legal, regulatory and administrative framework at all levels, strengthen regional and global technical support platforms for vaccine pharmacovigilance, make international expert scientific advice on vaccine safety issues available, put in place systems for appropriate interaction between national governments, multilateral agencies and manufacturers. So, monitoring of vaccine safety pre-licensure, so all vaccines first be tested and monitored. So, first they are tested in clinical trials. We have phase 1 which examines safety, dosage, side effects in small number of people. We have phase 2 studies examine vaccine efficacy and confirm safety and optimum dosage in large group people. Phase 3 we confirm that the vaccine is effective and safe by monitoring side effects and adverse reactions in a large number of people. As you can see here, this is the table, right. Now, pre-licensure studies, all this phase 1, phase 2, phase 3 are called pre-licensure studies. Okay. So, they identify common and acute undesirable reaction that occur with a frequency greater than 1 in 10,000 vaccination. However, we cannot detect uncommon or rare adverse event. We cannot detect in people who are having in particular populations like HIV positive, malnourished individuals, sensitive. So, we are not able to detect the reactions. So, as a result, post licensure exposure monitoring is very important to identify and evaluate such adverse events. Hence, the importance of effective vaccine pharmacovigilance is very, very important. Now, post licensure means identify rare reactions not detected during pre-licensure studies, identify risk factors that may promote reactions, monitor increase in the known reactions, identify vaccine safety signals that needs further study. It may be conducted by the National Pharmacovigilance Centers in collaboration with the WHO Collaborative Center for UPSLA for International Drug Monitoring, which is the UPSLA Monitoring Center. And they have succeeded, this particular centers have succeeded in achieving a great deal in collecting, analyzing case reports of ARIAs, distinguishes signals from background noise. Background noise क्या होता है? Coincidental occurrence, कोई ऐसे भी कोई adverse effects हो जाए जो vaccine की वजह से ना हो. Supporting regulatory decisions based on strengthened signals. Alerting prescribers, manufacturer and public to new risks of ARIAs. Now, what are the key pillars of vaccine safety pharmacovigilance? Passive vaccine safety surveillance means AEFI reporting to public health. Spontaneous reporting system is the major system, cornerstone vaccine pharmacovigilance system because of its relative ease of implementation, its cost, its ability to monitor all vaccines. They monitor events reported by healthcare providers, consumers, and they collect the data. Whereas the active means active measures are being taken. That is proactively soliciting information. Soliciting means collecting the information on adverse events from vaccine recipients directly or searching for events in clinical or administrative records. So, special studies, they also come under active vaccine safety surveillance. Special studies in response to signal detection or to collect additional information who have experienced serious adverse events following immunization. Now, in active surveillance systems, we have vaccines can undergo post licensure clinical trials, that is trials after the vaccine is in the market, okay, to assess the effects if there are changes in the vaccine formulation, vaccine strain, age, number and timing of doses, simultaneous administration, and try to take the interchangeability of vaccines from different manufacturers. To improve the ability to detect adverse events, which are not detected. So, this phase 4 surveillance studies, they are done on these specially health maintenance organizations. They are recruited from health maintenance organization, and the cohorts are the health professionals like pharmacists, nurses. Right. Then you have large linked databases. From there also the data is collected. Clinical centers, including clinical immunization safety assessment centers, tertiary clinical centers have been used to conduct the research. Now, what are the steps of vaccine pharmacovigilance? First of all is signal detection. Now, signal detection means we are detecting the signal that suggests that some adverse event for immunization is related to vaccine. That becomes a signal. Okay. Then we do the spontaneous reporting by healthcare workers through AEFI surveillance systems. Then we develop a hypothesis whether there is a possible causal relationship between adverse event and vaccination, and then we test the hypothesis through use of epidemiological studies. So, development of causality hypothesis, and then we do the testing of the hypothesis. So, the first component is detection and reporting, second one investigation, causality assessment, risk benefit assessment. Let us go one by one. Detection reporting. Who detects the ARIAs? Basically the parents of immunized infants, parents of children, health workers, the person who are working at the center, they are able to recognize. Health workers are responsible to detect the AFIs and report it, right. So, they have all immunization staff must be able to identify and report the adverse events. Detection requires effective staff training and education to ensure proper diagnosis. So, these are the health workers can report serious EFIs that have been caused by immunization errors, any events 30 days after vaccination, then swelling, redness, soreness if it lasts for more than 3 days. Then they conduct a EFI investigation because we need to confirm the diagnosis. We have to identify the specifications of the vaccine. We have to examine the operational aspects of immunization. We have to justify the search for other EFI cases. और कौन लोग हैं जिनको ये प्रॉब्लम हुआ है. Compare the background risk of adverse events, right? Then causality assessment, it is a systematic review of the data about the AFI cases, that means likelihood of a causal association between event and vaccine. So, causality assessment helps in if AFI is actually attributable to vaccine. So, what are the steps? Now, these causality assessment is done, they raise the awareness of vaccine associated risks, and this combined with knowledge of benefits of the benefits of immunization forms the basis of vaccine information for parents and vaccines. Now, quality of causality assessment depends upon the quality of the case report, effectiveness of EFI reporting system, quality of review process. So, 5 principles they help in do the causality assessment, like specificity of the vaccine, temporal relation, temporal मतलब vaccine के बाद कितने time के बाद adverse event आया था, strength of association, consistency, biological plausibility, and then we have to do the risk benefit assessment. Continuous evaluation of risks, continuous evaluation of benefits is required to strengthen the confidence in immunization program. The risk benefit assessment should address the population which is at risk, not the individual risk. We have to address the population risk. Take into account all type of issues, economics, availability of alternative vaccines and other factors. Be prompted by a duly identified risk. अगर कोई risk आए तो उसको monitor करना चाहिए और check करना चाहिए. And run in parallel, so active inquiry, cooperation and exchange of the information. So, these are the references of my topic, and thank you so much. I hope you have understood what is vaccine pharmacovigilance. So, it should be beneficial for all of you. Thank you.